reassemble it. And then
Alright, so I'm going to go ahead and give you this mic.
Oh, right.
And you can just put this in your pocket or wherever is comfortable for
that
you.
work?
Yeah, that's fine. And then here, maybe just midway, not quite that close, but maybe on your sweater. Actually, we don't have the volume control for the room. Is not adjustable, so yeah, just midway.
Does that work? Okay, thanks. Facial reconstruction of George Washington. Yes, I did. I recognize your face from somewhere.
Okay, from George Washington. Yeah, he and I are really good.
That's right. That's right. He scanned you too.
It was like on the History Channel,
Right.
PBS type special or
Right.
something like that. It was pretty cool. It was really interesting.
Thank you. It was a great, interesting project. I never thought I would learn so much about the 18th century.
Okay.
But how do I recognize your face from the talks? Or?
I've been to a decent number of these talks. I remember one way back where you mentioned how most mutations are recessive.
Yeah.
And I sort of intercepted you after how this could, they work their way through the system and could,
Right.
after a large mutagenic event, several generations down the line could create some sort
Right,
of
exactly.
for
And it can emerge as a novelty.
right. But then several generations down, they become like a almost punctuated equilibrium type, where a large number from a species start to.
Yeah, a number of individuals emerge with the feature instead of like one individual.
Yeah, but several, because presumably.
It spreads silently until
Because
there's
brothers,
critical mass.
siblings
Yeah.
don't mate, or
Yeah.
it takes a couple to get into the cousins type of.
Yeah.
Then all of a sudden they all emerge. I remember you said with laboratory rights and rats and mice, extra digits.
Yeah, they did this in, they did it backwards actually. They found, all of a sudden there was a litter of mice pups with extra digits. They didn't know where these came from because these are supposed to be genetically pure experimental lab strains. And then they backtracked it, and it turns out there had been a recessive mutation some generations before. It spread throughout the colony. You get a critical mass of heterozygotes, and then all of a sudden you get a bunch of individuals with the novelty. So it's a wonderful mechanism, and it doesn't invoke anything metaphysical or abiological or.
You can imagine if there's a large mutagenic event, like a radioactive asteroid that hits locally, or a volcano that kicks uranium or radioactive thorium or something, you could have an environment, a region where this would happen.
Yeah. You just have to sort of stress the cell's capacity to eliminate error.
Okay.
So whatever the stressor is, which will depend on the organism. A stressor for a mouse will be different than a stressor for you.
By the way, I have a very good memory, so it's not like I sit there.
No. Well, that's enviable, actually. Especially as one gets older.
So it's not like these two talks made such a powerful impression on my life. I remember these years later. I sort of remember a lot of it.
That's a blessing and a curse.
Which is a blessing and a curse.
Spent 19 years in a biology department.
But they were good. Good talks.
Thank you.
Thanks for doing that, Chef.
You here? How are you?
Very good. How are you doing?
Is there anything you want to say special in the introduction?
Whatever.
Set your own self.
All right, I think it's time to get started.
I play banjo.
I can mention that. All right, thanks for coming. Before introducing today's speaker, let me remind you that the next talk is on Tuesday at noon, and Andrew Buskell from Cambridge, who's a fellow at the center, will be talking, and right there, we'll be talking about cognitive novelties, informational form, and structure of causal explanation. You're all invited to attend Andrew's talk, and there will be no talk on Friday next week, and then we'll have a week without talk because it's a spring break. Today it's my great pleasure to introduce Jeff Schwartz, who is Emeritus Professor in the Department of Anthropology. He's a physical anthropologist who works on various topics. One of them is methods and philosophy of evolutionary biology with a focus on systematics. Another one is work on the growth of, I guess, the teeth and this part of the body, whose name I'm blocking on now.
Man.
Joe, that's right! And then it's, I'm almost American. A few more weeks and I'll be a real one. And the third area of research is the analysis of skeletons, particularly around the Mediterranean area. Also, he's been associated with the Department of History and Philosophy of Science for a very long time, and our students and ourselves have benefited tremendously from having him as a colleague. He's taught for us many times year-long seminars on the great books in evolutionary biology, and I remember some of my
Lots of complaints.
yeah, exactly what I was going to say. Big traumas. I need to read very difficult books for a whole year, but I think great memories as well. And today Jeff is going to be talking about molecules and evolution. And he plays banjo also. Jeff,
Thanks. Wow, great crowd. Thanks for coming here. And I'm glad you're applauding now. So anyway, the title had interbreeding in it, and that was only a way to sort of draw you in here. I'll get to it later on. So the real topic is molecules and evolution, and for decades I've been interested in the question of why are people doing what they're doing, especially in human evolutionary studies, which seems so different, especially historically, going back to the 17th century, from the rest of paleontology and comparative anatomy. So I've been adding a lot of new stuff, and I hope I can get everything in about 50 minutes. So I might talk a little more quickly. If I'm talking too quickly, please stop me. So I thought I would start with this comment from this embryologist I've become enamored of, Frank Leary, who wrote essentially, "We read the history of science very superficially indeed if we fail to recognize the constant interdependence of all scientific thought. Scientific discovery is a truly epigenetic process. Progress in each area depends on the movement of the whole science." And this is relevant because in terms of molecular anthropology, molecular evolution, and systematics, it's kind of been separated from morphology, and the question is, how can they be if they're both part of the biology of an individual? So I'm going to start with Darwin because that's actually part of the historical background to talking about these molecules and evolution. And it was in 1871 that Darwin took on the topic of human origins. He relied on the comparative morphology of everybody else. He didn't do this work himself, especially Thomas Huxley, the comparative anatomist. When it came to human origins, he basically started off with a premise that he introduced in The Origin and in his notebooks, which is that one would tend to find related species and their ancestors where you currently find living species. But the problem with humans, so if you have giraffes in Africa, you would expect to find fossil giraffes or elephants and so on. But the problem with humans is that humans are all over the place. And of the possible relatives, the three great apes, orangs, chimps, and gorillas, they're also far-flung. So the question is, how did he come to deal with that? Well, he ruled out Southeast Asia, the home of the orangutan, because it was tropical and it was just too lush. There weren't enough dangers to sort of have the selective pressures that would be necessary to provoke the emergence of something as wonderful as we are. But chimps and gorillas live in Africa, and we know in southern Africa there are lots of dangers and predators, and there would be the conditions that would allow selection to produce a human. So without any morphological or anatomical justification, he concluded that Africa must be the seat of human origins, and chimps and gorillas are our closest relatives. Now, there are some problems with this. Chimps and gorillas actually live in the middle of Africa, which is tropical. Southern Africa is thousands of kilometers south, and that's arid, semi-arid, and that's where you find the predatory cats and other things. And actually, if you look at the subtext of the volume 1 of the Descent, most people look at 2, which is sexual selection. The subtext is the evolution of civilized from primitive barbaric humans, and that's where you find supposedly primitive people in Africa. Now, he was preceded in 1863 by Thomas Huxley, who did the first morphological justification of Linnaeus's putting humans or classifying humans with other animals in a group that eventually became called primates. And even though people miscite Huxley as demonstrating that humans are most closely related to chimps and gorillas, the African apes, he did no such thing. In fact, at the end of the essay on man's relation to the lower animals, from which this illustration comes, he concluded that the great apes were a group, and perhaps somewhere in the distant past there was a common ancestor with humans. So effectively, the great ape group, chimps, gorillas, and orangutans, became the longest lasting alternative theory of who's related to whom amongst humans and apes, into the 1960s at least, with the work of Adolf Schwartz, who I've highlighted here. Now you can see there were other alternatives, and eventually one of these became more dominant, but at least for over 100 years, the human great ape theory dominated. So in the context of that, as fossils were being discovered, the first ape-like fossils were found in the 1850s in France, and other things were found in the late 19th century, starting in the Schwartz and in Pakistan. These fossils were plugged into this scheme, a human great ape group. In the 1930s, a PhD student at Yale, Lewis, who was sorting through the ape-like fossils that were coming from these Miocene deposits in Indo-Pakistan, discovered a jaw that he thought looked more human-like than ape-like. And by 1930, there weren't a lot of hominids known. The Neanderthals were discovered in the 1850s. We knew about things from Asia. There wasn't a whole lot from Europe except for Neanderthals. We only began to know about early South African fossil hominids in the 1920s. And there were various features such as a short snout, low cusp cheek teeth, and other sort of features that were brought forward in arguing why this particular specimen from the Schwartz and others that were subsequently found, named Ranopithecus, should be considered a human ancestor. So after some years of dickering about this, because it didn't sit well with various people, the thing that people settled on was that what really distinguishes hominids, humans and their fossil relatives, which is how I'm using the term, from apes and the others, is that we have thick enamel covering our molars. Whereas if you look at most primates, most mammals, there's thin covering of enamel on the molar teeth. And so that sort of linked Ranopithecus kind of stuff. Where's my dot? With hominids, Australopithecus, for instance, and things plunked into the genus Homo. So the scenario was that amongst the Miocene forms that were being found, like Dryopithecus from Europe and the stuff that I said from the Indo-Pakistan, there were thin enamel types and there were thick enamel types, and these were somehow, one of these was ancestral to thick enameled hominids, and you can see Ranopithecus is part of that potential ancestral group, as well as this thing Schwartz, which had originally been thought to be related to orangs. And even you look at teeth like from Lucy, Australopithecus afarensis, and like thick enamel. So that was okay. One thing that did sort of surprise people is with more specimens, they realized that these two genera, Ramapithecus and Schwartz, were really the same thing. And according to our rules and systematics, the thing that was the name that was presented first is the one that has dominance. So Schwartz was named in 1917 and became to take the place of Ramapithecus. But the problem with the scenario was that orangs had thick enamel. So if the great apes are a group and orangs are not closely related to humans, then how do you explain that? Well, orangs must have evolved thick enamel in parallel. And this is how people explain things all the time. They think they're generating a theory of relatedness, they're actually defending one. And therefore, when you have inconsistency, you have to explain them away. Well, in the 1980s, people started to find facial remains with the jaws of Schimopithecus, and lo and behold, they had a slew of the features that are otherwise distinctive of orangutans. The size here is wrong. This thing is actually half the size of an orangutan. But just to sort of point out the features, which I don't need to go into in detail. So this meant that if Schimopithecus is related to the orangs, it couldn't be related to hominids, as far as people were concerned, because they had shifted from the human great ape group more to the human African ape group. So if you embrace the human and African ape group, you couldn't accept that Schimopithecus and orangs were closer to hominids than the African apes. So you have to explain that away again. And this is when, as molecular studies had been gaining traction starting in the 1960s, which was part of the shift from the great ape to the human African ape group, then you got the dominance throwing out morphology for molecules, saying, oh, we molecular folks were right all along. Not only did humans not have a deep paleontological or geological past, they're most closely related to the chimpanzee, which totally contradicts all the morphology that I know. So in more recent terms, this sort of prioritizing of molecules over morphology has sort of taken over everything. And everybody sort of believes in this. A molecular biologist colleague of mine described this as the tyranny of the genome. And what a big group who's been getting a lot of news is Pablo and his group from Leipzig. So I'll focus a lot on what they're doing, which is giving priority over molecules. And this is what I want to sort of do right now, which is most of you have just heard about what they're doing in terms of claiming that there's a group called the Denisovans, based on molecules. They're somehow related to Neanderthals. This group is related to humans, and they all interbred. And in fact, from the molecules, they're claiming that there is a mysterious or a mystery hominid that we still do not know. Now, in terms of all that, I want to look at Heidelbergensis, Homo heidelbergensis, which is supposed to be then excluded from this group. Now, when I first saw the Denisovan material, which is where the name comes from a cave in Siberia, I thought, why are these things hominid? Anyway, here is one of the few specimens from which the DNA was extracted to sort of promulgate the notion of there being a Denisovan group and interbreeding between them and Neanderthals and humans. And what's interesting is if you actually look at the comparison of the Denisovan molar to a human molar, size alone, much less the morphology, is just like astonishing. I mean, that is like out of sight. And that's why it's important to think of the Heidelbergensis thing, because even in the reports on the Denisovan cave material, as well as the newer mandible from Xia, southern Tibetan plateau in China, people have said, wow, these Denisovans are so huge, and they're so primitive in their morphology. Isn't that wild? Because we know they're closely related to humans and interbreeding with them. So here are a bunch of specimens. I've studied basically the entire human fossil record, so these are my original slides. So if you look at Neanderthals, and the star is where the third molar is, which is the counterpart of the Denisovan tooth I showed you. Here is Homo sapiens, Neanderthals, another Neanderthal, specimens that are allocated to Heidelbergensis. Very small third molars, very minimal morphology. If you look at the Denisovan upper molar, it looks like an Australopith. And these Australopiths are these big, chunky, early hominids that we know from South and East Africa. And in fact, if you look at the articles, they say, look how primitive these Denisovan teeth are, in spite of the fact that we think that they're more closely related to us than Heidelbergensis or anything else. So the question then is, what makes molecules dominant over morphology, or why not both of them together in terms of trying to solve some of these problems? So that's where looking at the history of these ideas is actually very important. So the first monograph, the first big study that got into the molecular side of these things was published by this British bacteriologist, Nuttall, And basically the idea is blood's thicker than water. And I think we can sort of relate to that because my relatives are closer to me and there'll be more in our blood together than, someone distantly at the back of the room. So his idea was, because immunology was becoming a thing at the turn of the century, was let's look at blood serum proteins because the relationships are in the blood. So what you do is you take in this case it was whole blood serums instead of breaking out the molecules, and you put them into a host, like a rabbit. The rabbit produces an antibody. You take that anti-serum, you put it back with us, and there will be perfect bonding. So you get this big precipitate out in the test tube. And if there's less bonding, then you get less precipitate, and so on. So the idea was that the more precipitate, the more bonds in common these things had, the more similar they were, the more closely related. So what they did was they looked at and that was his idea. So I skipped ahead. That sort of fell into limbo into the 1960s, and in a volume called The Biochemistry of Hemoglobin, Zucker, Condell, and Pauling published this article which sort of had this throwaway thing about evolution in it. And they did this hemoglobin, anti-hemoglobin. Hemoglobin is a metabolically active protein in the serum. Thing, and they used fish, which is weird because fish bind oxygen differently, chickens, horse, and gorilla, and human. And in terms of the hemoglobin, anti-hemoglobin tests, it seemed to follow a pattern of from increasing similarity going to gorilla and human, which conformed to more or less the morphological take on how these animals were related to one another. So then they said they had to try to explain how this happened. So what they said was, overall similarity, as demonstrated by this, must be a reflection of evolutionary history, with descendants mutating away from each other, becoming gradually more different from each other. So the point of all this is that molecules are constantly changing. So the more similarity you have is because you've shared a longer history of the same changes through ancestors, and have less time to diverge to accumulate your own differences. And this is what I call the molecular assumption, and this is still how people proceed in terms of working out phylogenetic relationships, or claiming to, from the molecules. So let's start with some nuclear DNA before we get to mitochondria, and this probably is too simple for everybody, but there you go. So the notion that people still, I think a lot of people still embrace, and this is something that Paul Griffiths and Carola Stotts and others were talking about, which is the genes for idea. And basically the idea is that you have a sequence of these nucleotides. It used to be that people didn't know that there were spacers in nuclear in sequences that are called introns. Introns really don't mean anything. They might have an embryological function, at least as far as people think. And so their size doesn't mean anything. They're only spacers between the things called exons. And then RNA would sort of get rid of these spacers, and then you would have a sequence, and that would be directly translated into a protein. So it's basically one gene, one protein is the idea. So the question then is it relevant to compare DNA sequences? Well, if you're a bacterium, yes, because in bacteria the sequence means something, because 97 to 98% of the bacterial genome codes for metabolically active enzymes and proteins. So if you change one of those nucleotides, you're going to change the amino acid, you're going to change the sequence that creates the protein. But all this does is allow bacteria to adapt. They don't evolve. Millions of old fossils of bacteria, the rods and the spirals and the stuff, look the same now. So what's really happening is that these things are just sort of adapting because they produce millions of themselves from generation to generation. And statistically, some of them will be able to survive in one situation where others don't. So they're not evolving, they're changing, but change and adaptation and evolution are not necessarily all synonymous. In metazoans, multicellular animals, it's the reverse. Only about 2 to 3% of our genome is coding, and the bulk of it is non-coding. And this is important because that bulk of it is intimately tied in with our development. Our becoming, what's really interesting about being a metazoan or a plant is you have topology, you have shape, and you have complexity. So how does this work in metazoans? Well, it's been known now for some decades of this thing called alternative splicing. And here, really, DNA is dumb. You know, this 97 to 98% is just lying there, waiting. Sorry, I don't want to impose that. And RNA comes along and starts using that sequence to produce proteins that are relevant at that particular developmental moment. So you can, the introns or the pink things, they could be spliced out, and you can have RNA sort of putting together these different components in all different ways, including reading the other way. So reading that way is called sense, and reading that way is antisense. So you can have RNA sort of going all over the place, clipping together these different chunks of DNA, producing these different temporary proteins that are used in development. So an example is in the chick's inner ear, or cochlea, there are about 500 proteins. And a protein is a crystal, so they sort of resonate to a different frequency, which is, you know, they're in the cochlea. So according to the dogma, there should be 500 proteins. Well, there isn't. There's only one so-called gene, one expanse of DNA sequence, which is identified by a thymine-adenine sequence at the beginning of it and at the end of it. And RNA comes along and it recognizes one end. It says, this end says, start reading here. And then RNA goes in there and chops these things apart 500 different ways to produce the 500 proteins that are in the chick's cochlea. So it's that kind of alternative splicing which makes it possible to have actually not a lot of genes, but to be a complex organism because of the complexities and the infiniteness of alternative splicing. So the thing is that in metazoans, the formation of the organism is a tightly controlled sort of choreography between development of the regular genes, transcription factors, and a whole bunch of other things to produce this organism. So derailing that actually is not a good thing to do. And there are all sorts of mechanisms, especially with this class of proteins that we have called stress or heat shock proteins, whose jobs include keeping change from happening. So molecules are not constantly changing. They're actually supposed to not change with heat shock protein intervention. So let's go back to the Leipzig group, and they're making all sorts of claims about how you can you create a human morphology from a Neanderthal morphology and so on. And we'll talk about this one autosomal gene, the EVC2 gene. And when it's mutated, some individuals develop a large pulp cavity called taurodontism in their molars. And we humans have small. So Neanderthals have large ones and humans have small. So Pablo and his group said, well, what if we were to change the EVC2 gene, we could create a human molar from a Neanderthal molar. Well, when you change EVC2, and it's relative, we don't know how it's related on the same chromosome, you get a bunch of different syndromes. And the common feature of all of them is dwarfism. So here's a list of things that happen when you dysregulate or interfere with the proper functioning of the EVC2 and EVC genes. And one thing that happens is that humans will develop taurodontism. But basically, if you start messing around with these genes and their signaling, you get a lot of bad things. So there's something else about the way in which this molecular stuff is being used, which is assuming that Neanderthal morphology is primitive relative to human morphology. So the taurodontism, because it's in the Neanderthal, should be the primitive condition from which human small chambered molars emerge. In fact, taurodontism is very rare in mammals. If you look at hypsilon animals that grow their teeth throughout their lives, like horses, then you get big pulp cavities. But in terms of mammals in general, taurodontism is about as rare as the Neanderthals having So, and the other thing is to bear in mind is this is not cherry picking. You can't cherry pick what you want out of these problems, because when you muck around with the EVC2 gene, you get dwarfism, no matter what else you get in company with it. And we know we have a large Neanderthal fossil record. There are no dwarfs, and there are no individuals that are showing EVC anomalies. So another example that they wanted to sort of use from looking at the sequences is they identified the RUNX2 gene transcription factor in the Neanderthal DNA sequence. Well, in fact, that sort of should not be a surprise because the RUNX2 signaling pathway is relevant in all vertebrates for the normal development of teeth, bone, cartilage, and so on. So what they suggested was, since in experimentation and in normal clinical cases of dysregulation or interference with the RUNX2 signaling pathway, you get changes to the skull, big bulbous head, and to the rib cage and to the clavicle. And we know that the humans have this big bulbous skull and a small face, and Neanderthals have large clavicles and barrel-shaped rib cages compared to us. So the idea is that you would somehow take a Neanderthal morphology, muck around with RUNX2, and create Homo sapiens, and you can see in this boy here the way in which this Neanderthal is supposed to have morphed. Again, they're assuming the Neanderthal is the primitive condition. On the other hand, when you dysregulate a lot. And this disease is called, or syndrome is called cleidocranial dysplasia, cleido meaning clavicle, and cranial referring to the skull, and dysplasia means it's not properly formed, it's not fully formed. And there are lots of anomalies that occur with dysregulation of the RUNX2 signaling pathway, which means it has to remain stable for vertebrates to be functioning organisms. So there are a lot of the complications that arise with dysregulating RUNX2. The ones in blue are important because I'm going to show you a few specimens that I've identified this syndrome in a minute. But basically the same thing with dysregulating EBC and EBC2. You get a lot of bad things. And you just don't get one, you get a bunch of them. And some of these things are going to be lethal. If you get skin lesions, imperforate anus, you have pathways for infection. Incorrect insertion of the tongue, you can't swallow, you might be able to eat properly. You get incomplete fusion of the auditory tube, the bony tube in your ear, which allows for deafness, which is associated with deafness in an area if you have damage there for infection. And so in mice that are experimentally manipulated to dysregulate the RUNX2, they get the same morphologies as humans, which we know from clinical cases that have RUNX2 problems. And interestingly enough, when I was starting the Human Fossil Record Project in the 90s, there's this little skull from a site in France called Pécharazet, and everybody said, look how weird this thing is. And when I was describing it, I thought, look how weird it is. But it turns out this is very important because it has all the features of RUNX2 dysregulation or cleidocranial dysplasia. We don't have the clavicles, but we have the skull. And features are retention of the superior fontanelle that we see in fetuses that usually close over, and it's huge. This is not broken edge. This is real bone. Some of it's broken, but most of it is not. And you get retention in the fetus, like in all mammals, we have right and left frontal bones. In most mammals, they don't fuse until old adult. In primates, they do fuse early on. But it's considered to be something to look at if you find a patent metopic suture in humans. And this is what we find in the Neanderthal specimen. So this has the features associated with cleidocranial dysplasia. And it's got this tiny little face, and I'll show you some other things. The other thing is that I discovered this in a fossil Homo sapiens child, a few thousand years younger, from Russia. And you can see this part is all damaged, but you look under a lens, this is a good edge, so that's part of the fontanelle. Here's the patent frontal suture, just like we saw in the Neanderthal. Here are really sort of an anomalous dental morphology, which is part of the RUNX2 dysregulation. And this is a photograph of their bony auditory tube. And this is basically the black area is where it's unossified. So cranially and dentally, they have the features of cleidocranial dysplasia. And what this points out is when you dysregulate RUNX2, you don't get a human. You get something with RUNX2 dysregulation, cleidocranial CCD. So here you have a Neanderthal with CCD, and here you have a Homo sapiens with CCD, just like you do in present day. So in other words, if molecules were constantly changing, as the molecular assumption would have us believe, and these folks are using, is that you would have just chaos, developmental chaos all over the world and over the place, including in all vertebrates. You wouldn't really have normal vertebrates. All right, so what about mitochondrial DNA? Well, it's widely accepted that, in contrast to nuclear DNA, which has contributions from both parents that combine upon fertilization, the mitochondrial DNA, or the mitochondria, which contain the mitochondrial DNA, comes solely from the mother. Well, this is sort of like jumping ahead because this is sort of what the punchline is going to be. First of all, it always boggles my mind that people put mitochondrial DNA and nuclear DNA in the same analyses because they're totally different animals, so to speak. Mitochondrial DNA takes the form of a bacterium, and there's this long history of saying, where did mitochondria come from? Was a symbiotic relationship with bacteria that then got sort of embedded. It's a circular chromosome. It's only one. It means that it can be copied completely, whereas in linear chromosomes, which characterizes nuclear DNA, only one strand can be copied completely, and that's why we die, because the telomeres are constantly being eroded, and we eventually die. But these things are essentially immortal, if you want to think about that. They're basically involved with the cell's respiration, the energy of the cell. It was suggested, it's now becoming clear that nuclear DNA has repair mechanisms, as I mentioned, with stress proteins. It was thought that mitochondrial DNA does not. It's now becoming, there's some evidence that they do have repair mechanisms, so if there's a mutation in there, it doesn't necessarily stick. Mitochondrial DNA is increasingly being demonstrated as being inherited from the father, not just from the mother, and that there is recombination of mitochondrial DNA. Somebody had even suggested that we might start talking about mitochondrial Steve in addition to mitochondrial Eve. The notion of a D loop is based on the idea that there's this rapid evolution. Where does it take place? So it has to be in some area, and that's been called the D loop. That's not necessarily real. And there's not one mitochondrial DNA genome. In fact, not only do different cells have different mitochondrial DNA of different lengths and sequences, but you can have them within the same cell, which is called heteroplasmic. So there is not one mitochondrial DNA sequence for Homo sapiens, for Equus, or whatever. So where does this notion of inheritance come from, the notion of mitochondrial Eve? Well, it's really sort of interesting history, and I sort of had to stop myself from doing this so I could come here and give this talk. So there's more things that to be discussed and for me to learn. Now, fertilization was actually observed in the 1890s, but the first publication showing this was in 1895 by E.B. Wilson, who some of you may know from his work in the 1920s and 30s on genes. He was the first person to use the word gene, but he misspelled it as gen. In any case, so he chose an animal where visually he could photographically represent the egg and the sperm. And that was this sea urchin Toxoneustes variegatus. And he chose it, as I said, not because it's typical, but because he could visualize, he could photographically tell you what was going on, and the process of fertilization was slow enough that he could actually photograph it. So he describes the sperm. They differ wildly in appearance from the egg. They're very minute. In most cases, they have a long, vibrating tail by means of which they swim around rapidly. And fertilization, as had been demonstrated a few years earlier, is the combination of the sperm and the egg. So the illustration that he gives, primitive as it might be, of the sperm is really interesting because he recognizes that the head includes two parts. There's the nucleus, which contains the DNA, the nuclear DNA, and there's the middle piece, and then there's the tail. So in the sea urchin, what happens is that the entire head, the middle piece and the nucleus, goes into the egg and the tail does not. And he describes this. This is the first real description of fertilization, which is when the head of the sperm touches the cell membrane of the ovum, it activates the process of fertilization, which was actually better demonstrated and described by Lilly, and I'll talk about in a bit. So what happens is the head touches the egg responds, this cone sort of emerges, the head disappears into the cone, the egg produces this membrane, the vitelline membrane around it, which contains the tail, and that is sloughed off and the tail disappears. Now, that is the case in the sea urchin. But as he points out, in many cases, however, in the axolotl, which is an amphibian, earthworms, butterflies, and other animals, the tail also enters the egg. And here, I'm sorry, that's Nereus. That's the sea urchin. So basically, the tail staying out of the egg is not necessarily the typical case. All right, I just said that. Now, for mitochondria. Mitochondria were discovered in the 1890s, so people knew about their presence in eggs and in sperm. And the person I quoted from at the beginning, Lilly, is just a fascinating person to read. And he published this book in 1919, where he really is the first, he's the father of understanding fertilization, if you wanted to get into that.
You lost your image. You want to wiggle the cord, I think.
Okay. So he has a more complex diagram of sperm. But basically, the elements that Wilson had diagrammed are there. So you have the nucleus. This part up here is the preparatorium. The middle piece, that's the important part, and then you get the tail. And he then talks about, as others had, the mitochondria are in the middle piece. So that's really what you have to pay attention to. Now, he chose a worm of the genus Nereus for reasons analogous to why Wilson chose the sea urchin, because you could visualize what was going on, and because the process of fertilization was so slow, you could photograph it as well, and that's what he wanted to do. That's what I just said there. So then he describes this. And he talks about similar things happening as did Wilson. The head touches the egg reacts, produces this cone, the head disappears into the cone, and the rest is sloughed off with the vitelline membrane that the ovum also produces. In the case of Nereus, it's only the nucleus part of the head that enters the egg. The rest is sloughed off. So he describes this, but he says the case with Nereus is exceptional. He only chose this organism because he could visualize the sperm in the egg, and because the process was so slow he could photograph it. This is exceptional. In most cases, the entire sperm enters the egg, including in nematodes, like ascaris, crustaceans, mollusks, insects, amphibia, and mammals, and some sea urchins, which is what Wilson described. And this is from Lilly, sort of showing cases of different animals where the entire sperm is in the egg. So, consequently, it seems that the typical case of fertilization in animals is for the head, nucleus plus middle piece, to get into the egg, including the tail in a lot of cases as well. Now, he cites this German author, and I've only gotten the references. I'm waiting to get the articles, but he translates them, which is a good thing. In ascaris, which is a nematode, this entire sperm enters the egg, and the mitochondrial granules flow out into the egg, and they become intermingled with the egg's mitochondria. And in fact, what's really interesting, because this is, people are now trying to defend maternal inheritance, is that he can follow the mitochondria, because mitochondrial DNA was not analyzed then, through different cell divisions. It existed into multiple cell divisions in the organisms, which means that it's not eliminated, that it and its contents are not eliminated at the beginning, which would be the premise for maternal inheritance. So from these cases, it seems to me that we can say that the typical case, or at least not the rare instance, is in animals for the at least the head, the nucleus, and the middle piece, if not entirely the tail as well, and the mitochondria to get into the egg. So how did the opposite become fact? Well, it's very interesting, because if you look at the history, this is fascinating, and it's H.G. Wells, which makes this even more interesting. So H.G. Wells had written all sorts of great things. He had written this popular history for people. It was so popular, he became this very wealthy guy. He wanted to do the same thing for biology. And so he hired Julian Huxley, Huxley's grandson, and you probably know of Julian Huxley from development and from notions of scaling. And his son, George Philip Wells, who was a physiologist. Now, H.G. Wells had studied zoology, with Thomas Huxley. He was, in fact, his lab assistant. But he never did anything scientifically except in 1893 published a handbook, a textbook, primer for exams for biology students, the text of biology, which went through multiple reprintings but never any revisions. So basically the writing of the Science of Life was done by Huxley, Julian Huxley and G.P. Wells. So this originally came out as a series, chronicled series like National Geographic or whatever, between 1929 and 1930, and in a 3-volume work. In 1930, it came together as a single work with 9 books, which were the different divisions of the original publications. In English, it was published in various languages, but I'm just interested in the English language, in 1934, and the last one was in 1939. Now, if you compare the British and the American editions, their pagination is different. Some are 2 columns, some are no column, full page. But the language is the same. The words are identical, and the page numbers may be different because of the way it's typeset. So basically, they give the interested popular audience, here's a sperm, here's the ovum, this is what they're all about. The sperm are the male gametes. They have a head and they have a long, lashing tail, which it can use to propel itself around. And this kind of sounds like the language from Wilson's, the sperm having this vibrating tail that it swims rapidly about with. Now, what's really interesting in the illustration that I just showed you is that only the head and the tail are emphasized. And in fact, if you look at the figure legend, basically it's emphasizing the shape of the head, which has already been described in the text as globular or bulbous. So you basically have the head and everything else. And the impression that I think this leaves people with is that there's a head and a tail, and that's it, in spite of the fact that the illustration also shows the middle piece that Wilson and Lilly and other good scientists were interested in. So then they talk about what happens when you have this army of sperm floating around, swimming around, and it comes into contact with an egg, propelling themselves by true paths. It sounds like Lilly's depiction of these free-swimming sperms sort of running around in these circles or spirals, beating its tail. They're correct about fertilization. The act of fertilization begins when the sperm presses its head against the egg. That's what Wilson and Lilly were described. But then there's this interesting thing that they say. After fertilization, the sperm burrows its head into the substance of the ovum, leaving the tail outside to wriggle for a while and then to perish. This is the climax of the discussion, and I thought that was a joke. And that's the end of the statement. So, where did they get this idea? I don't know. It seems possible to infer that they were channeling Lilly in its work on the worm Nereus, where only the nucleus part of the head gets into the egg. But that's about where the comparison ends, as far as I can tell. Now, Lilly did publish a 1923 edition of the book, but the words, the language on fertilization are identical between the 1919 and 1923 edition. So where did they get this idea of wriggling and then perishing? Especially because the literature that they could have read talked about things being sloughed off that are captured in the vitelline membrane that's produced by the egg. The other thing that's interesting about the Science of Life is that there were no references in it. None whatsoever. So the idea is, where do these things come from? So as I said, the only scientific thing that H.G. Wells published was his textbook in 1893. His son was a physiologist who was actually interested in the mucus of various invertebrates. He published one article before the publication of The Science of Life, had nothing to do with fertilization. Huxley cites Lilly, but way afterwards in Huxley's famous book on the problems of relative growth, has nothing to do with fertilization. So basically what I'm thinking is, all right, so Huxley and Wells Jr. were lecturers in zoology. They may have read Wilson and or Lilly, and they got it wrong. And this sort of ended up in the textbook. But how did this become sort of common knowledge, so to speak? Dumb luck, whatever. Interestingly, the book Science of Life was used in the U.S., actually, I forgot to put that in there, in textbooks into the 1960s, and mitochondrial DNA was discovered in 1964 and 1968 through two different techniques. So at some point, the notions strict maternal inheritance and clonality, because if you're a single thing and you're not recombining, you're just cloning yourself, became the mantra that's used in phylogenetic analyses, especially in human evolution. So the folks who were sort of tagged with the naming mitochondrial DNA Eve, or mitochondrial Eve, were this biochemist from Berkeley, Alan Wilson, and his students at the time, Becky Kahn and, I forget his first name, but that's okay, he's forgettable, Stone King. So in 1985, before the mitochondrial DNA Eve thing, they published this thing about, it would be nice if we knew some sort of reliable properties about mitochondrial DNA. If it were true that the sperm contributes no DNA to the next generation, and mitochondrial DNA within the egg is homogeneous, this would be a great tool. This would be great for us to use, because there's no recombination, there's no sort of ambiguity introduced by fathers, but we don't have proof. 2 years later, they write, because mitochondrial DNA is inherited maternally and does not recombine, it is a tool for relating individuals to one another. Where is their reference? The reference is to this Olivo paper from 2 years before the previous one that I commented, and they said, basically they say, it is thought to be related to strict maternal inheritance. So somehow the language became definitive, that this is strict maternal inheritance and the implication is clonality. But there's been a parallel history of people questioning this, including this wonderful molecular biologist, Ira Walker, who said what is perhaps most surprising is how strongly evolutionary biologists have believed in the strict clonality of mitochondria. It is not clear whether this faith arose through wishful thinking. After all, there's so much we can infer if this is true. And the belief that a little recombination would have few consequences, or the perception of a misconception through the tangled web of article citation. So essentially, repeating this 1,000 times doesn't mean that you're demonstrating it 1,000 times. And as recently as 2017, one of past co-authors with Ira Walker, Ladoukakis and his colleagues wrote, the history of our understanding of animal mitochondrial DNA is itself a lesson in how we gain knowledge of the complexity in biology. In the early 80s, it was a very accommodating molecule, God's gift to those interested in phylogeny, taxonomy and population genetics, small, conservative in gene arrangement, uniparentally inherited, homoplastic, meaning they're all the same in every cell, non-recombining. Gradually, however, the molecule presented us its true face. Size is valid, but there's large variation. So that the mitochondria within the same cell can be of different lengths and of different sequences, not just between cell types. Maternal inheritance and therefore homoplasy can be bypassed in many ways. Recombination occurs in many ways. It's anything but a single-purpose molecule. The future of this research must be in the role of the function of the organism, and its value as a tool in the study of novelties in the history of life. In fact, one of the best cases of demonstration of paternal inheritance, which is called paternal leakage, and paternal-maternal recombination, and of heteroplasmy, comes from humans, which was published in 2002. All right, so to wrap this sort of up with the phylogenetic stuff, what do we do? What's going on? So here we go. This is the interbreeding part. It's going to go very quickly. So the idea is that because of these genetic analyses, there's this group Denisovans and the mystery hominid, which is not put in here. Neanderthals are supposed to be related to them, and everybody is having a great old time in their Paleolithic orgies. Now, there's interesting things here. First of all is the notion of genes, that there are Neanderthal genes, there are sapiens genes, there are Denisovan genes, and so on. Effectively, what they're talking about are SNPs. These are not genes. These are single nucleotide polymorphisms, which means that they're just a single base in a whole nucleotide sequence. And you can have alternative states between pyrimidines and so on. They flip back and forth. That's fine. But those aren't genes, and those alternative states are not alleles. These are just little bases in a sequence. Now, there's something else here, is that this whole phylogenetic tree thing is assumed. It's not generated from the data. So it's assumed that chimps are primitive relative to the rest, that Neanderthals are closely related to humans, and then the genetic stuff is mapped onto it. Now, the other thing is that Pan, the chimp, is considered primitive. And what's defined as sapiens, Neanderthal, or Denisovan, or some mixture, is by their differences from the chimp sequence. But if you go to the molecular assumption, everything is changing. So you can't eat your gene and have it too, or whatever the phrase would be, where the chimp sequence doesn't change and everything else does. Either everything is changing because it's the molecular assumption, or nothing is changing. And again, as I pointed out, if nuclear DNA was changing just ad libitum, there would be death all over the place. Now, the other thing is that these aren't the only hominids that existed. As far as I'm concerned, and even some of my more conservative colleagues, there are various other possible species of hominid. And the relationships demonstrate that humans and Neanderthals are not necessarily the closest of these hominids. So I want to get back to that group Heidelbergensis that was kind of like pushed to the sideline because of the priority given the Denisovan DNA. There's a whole bunch of specimens. I'm not so sure this is just generally. These are a whole bunch of things that are considered to be Heidelbergensis things. There's a bunch of them. And they're different, and some of them have to be different species because their morphology is so different. And then there are a bunch of things that are closely related to Neanderthals that are not Homo sapiens. And there are probably a few more things as well. So there's a Heidelbergensis group, or clade. There's a Neanderthal clade, and my recent work has been especially on what are these fossils called fossil Homo sapiens. A lot of them are not. And we can have that discussion later if you want, but this is just a sort of sampling. There's a sapiens clade. So there are things that are more closely related to Homo sapiens to us than to anything else in the human fossil record. And what's interesting is morphologically, one can make a better case for uniting the Heidelbergensis and the Neanderthal clades than one can for uniting a sapiens clade with either. Doesn't mean that that's not the case. But morphologically, this is a stronger case to make than uniting humans with a Neanderthal clade. Even if one did, you can see that Neanderthals and Homo sapiens are not very closely related. They're not like that. So basically, until more specimens, including so-called anatomically modern humans, are sequenced, you can't assume that the similarities between humans, Neanderthals, and these Denisovans, or between any of them, are unique to or you derived for them. That these are actually specific features of either a Neanderthal or a Homo sapiens. They could just be primitive retention. There could be all these different species that we have not and maybe will never be able to sample that have the same sequences. So it doesn't necessarily mean if you find them here that they mean anything at the phylogenetic level. Another thing is that similarity also in cladistic analysis can be no change. So the idea that molecules are constantly changing, which kind of violates the biology of stress proteins keeping this from happening, similarity could just mean no change whatsoever. And you cannot assume that Neanderthal morphology is primitive relative to humans. So what about interbreeding? Well, the same kind of thing. We need to know more samples from different hominids. We will never get them. A lot of these so-called anatomically modern human fossils don't preserve any DNA whatsoever. They're now turning to proteins, which I think is the same problem, but we can talk about that later. But what's interesting is just a couple days ago, I started to find studies on hybridization between different groups or different species, and it turns out that in the studies on this hybridization, that it increases the likelihood, increases actually the event of paternal leakage and paternal-maternal recombination of mitochondrial DNA. So if Denisovans, Neanderthals, and Homo sapiens were interbreeding all the time, you would have this mitochondrial chaos, which is not what the model is suggesting to us. You also just can't generate theories of relatedness among hominids from looking at chimpanzees alone. You have to at least look at all apes. And as far as I'm concerned, you should at least look at monkeys as well. So you get an outgroup comparison. You might as well look at other mammals, too. So some closing thoughts. What's interesting is, to me, is since the molecular assumption was originally validated by general congruence with the morphologically based relationship, morphology should falsify the molecular data when there's a incongruity, not the other way around. The other thing is that molecular anthropologists and people who buy into this are continually promoting the idea that there is some difference between molecules and morphology. Molecules get priority of morphology as if they were distinct entities, but they're not. They're all part of a developmental continuum. Morphology exists because of the long chain of interaction between regulatory molecules, proteins, and so on. In other words, morphology is a reflection of the genome and the epigenome in action. Consequently, in spite of the appearance of things being sorted out, I think we're still at the beginning of understanding this biological realm and the relationship of the origination of morphological novelty. And we can't afford to presume otherwise and give priority to what has become received wisdom. So in the words of the philosopher George Patton, if everyone thinks the same way, no one is thinking. Thanks.
We'll take a 2-minute break, and then we'll reconvene for questions.
I'm okay, how are you?
Yeah, it's getting much better. Yeah, it takes time, but it's getting better. There's a few more time.